transcription chapter, do you feel that transcription is not a really very clear transcription or application? If you feel like that, I will go one day with a short revision of this chapter, if you want. Okay? Okay. So let's start with this one. Do you understand this part? One gene, one engine hypothesis. Okay, I'm giving you a little description about this one. So the first thing is that we need a minimal media. This is called the minimal media, and another is called the complete medium. Okay. So what is complete medium? Complete medium means everything is there, so the neurospora can grow on that. Everything is here. All the nutrients are there, so they can grow very easily. This is called the complete medium. What is minimal medium? Minimal media is something like that. More or less necessary things are there, but they need to synthesize little more things by themselves for their growth. That is called the minimal medium. Okay, more or less everything is there, but a little bit less. Very essential things are there, but not everything. Okay, that is called the minimal medium. Okay, and for the growth, what neurospora needs. So if I put them in minimal media, they need arginine to grow. And then there is growth. Okay? So they need arginine to grow. That is the main thing. If it is complete media, arginine is already there, so there is no problem for them. Okay? But if it is minimal media, they need to synthesize arginine by themselves. Clear? They need to synthesize arginine by themselves so they can grow. Here, arginine is not here. Not arginine. No arginine is here. So they need arginine. They can synthesize arginine by themselves from this minimal media to grow. Okay. But this process is a series of biochemical process. Something is there. Suppose this is not one-step process. Something is there. There are two medium steps here. Then the arginine is here. Okay. So from minimal media, they need to produce this substance first. Then this substance, then they can produce arginine, so they can grow. This is a three-step process. Okay. As it is biochemical reaction, that means each and every step is controlled by one enzyme. So one enzyme is required here to produce this product from minimal media. Another enzyme is responsible here to convert this product into this product. Another enzyme is required here to convert this product into arginine. Clear? So three enzymes are required for this total process. And there are three steps. We don't know what are those steps. Okay? Now the thing is that we prepared a couple of mutant stains. So here is neurospora. You irradiate with X-ray. If you irradiate with X-ray, it can create some kind of mutations. Okay. Now suppose in one gene there are one chromosome, there are three genes. If you put X-ray, it can mutate this one, it can mutate this one, it can mutate this one. I don't know. Okay. So I took three groups of neurospora here. Three groups of neurospora. All are X-ray irradiated. That means all are mutant groups. But I don't know what are those mutants. I don't know. I have no idea. Where is the mutant in A, B gene, B gene or C gene? I don't know. Now, how can I know that? and that is related to that thing. Okay. So how can I know that? Now the question is that I put, there are three things: ornithine, citrulline, and arginine. Three things are there. Okay. In the first group, I put arginine. In this media, I put arginine. They need arginine to grow, so they will grow. Because arginine is required, so they will grow. So if I put arginine here, they will grow. If I put arginine here, because that is the end product and that is the main thing, they will grow. If I put arginine here, they will grow. So all three, their main thing is that arginine. If arginine, I can put arginine from outside, they will grow. Okay, that is the main thing. That is gone. Now I put citrulline here. They can grow. I can put citrulline here. They can grow. But I put citrulline here in this group. They didn't grow. What does it mean? They can grow in arginine. If I supply arginine from there outside, they can grow. But if I put citrulline here, they cannot grow. What does it mean?
Does it mean that they can't synthesize the citrulline?
They cannot synthesize the citrulline. That's true. That's true. But why they didn't grow?
Citrusine inhibit arginine from producing?
No, that's not the thing. Three things are required. One is ornithine is required, citrulline is required, arginine is required, and the process is that if I put minimal media, they can, from minimal media, they can produce ornithine, they can produce citrulline, and they can produce arginine as a three-step process. This is a chain process. Okay. If one thing is not there, another thing will not be produced according to this model. Okay. If ornithine is not here, citrulline will not be produced. If citrulline is not there, arginine will not be produced. Okay. If minimal media is not there, then ornithine will not be produced. Okay. But I am supplying citrulline from outside here. But they didn't grow. Why? What does it mean? When I am giving arginine, whether there is citrulline or ornithine is there or not, that doesn't matter. When I am giving them arginine, they are growing. That's not problem. Because they need arginine to grow, I am giving arginine to them in the minimal media and they can grow. And they are mutant, all the mutant. They are not normal. So they cannot synthesize, as they are mutant, they cannot synthesize in this whole process. They cannot synthesize it. That's why they are called mutant. I am giving citrulline, but they cannot grow. What does it mean? I am giving citrulline, but they can grow. Just think about that. I am giving citrulline to their media, they can grow. What does it mean? Citrulline is not the main component for their growth. Main component is arginine. Okay. But if I am supplying citrulline from outside, they can grow. What does it mean?
Does it mean that they can not only grow with arginine, they can also, it's something maybe in their makeup that they can also grow with citrulline?
Not like that. They need arginine to grow anyway. They need arginine to grow. Whether you need to supply from outside or they can synthesize by themselves. The end product is arginine. That means if I am giving citrulline and they cannot grow, if I am giving citrulline and they can grow, that means when I am giving citrulline to them, they can synthesize arginine from citrulline. So this enzyme is active for them, so they can grow. They can synthesize arginine by themselves, so they can grow. Okay. If I am giving citrulline but they cannot grow, that means this enzyme is defective. They cannot change, they cannot convert arginine from citrulline, so they cannot grow. So this enzyme is defective. It cannot synthesize arginine from citrulline. Clear? Okay. Now the question is that when I am giving citrulline, they can grow, but when I am giving ornithine, particularly for this group, in ornithine they can grow, ornithine they cannot grow, ornithine they cannot grow. What does it mean? Ornithine cannot grow, ornithine can grow. I am giving ornithine, they can grow. That means when I am giving ornithine to them, they can synthesize citrulline from ornithine, from citrulline to arginine, so they can grow. Ultimate result is arginine to growth. So they can have this whole process, so they can grow. Clear? That means their enzyme 2 is working, their enzyme 3 is working, so they can grow. When I am giving ornithine, but they cannot grow, what does it mean?
Enzyme working.
That means ornithine to citrulline, this enzyme is not working. I don't know whether this is working or not, because if there is no citrulline, whether it is working or not working, that doesn't matter. At first, I need this step. I need citrulline from ornithine, and this enzyme is not working. If there is no citrulline, whether this is working or not, that doesn't matter to us. So I need to give ornithine when I am giving ornithine, and they cannot grow. That means this enzyme is defective, enzyme 2. Why do I need to give them ornithine? Now the question is that they are mutant, they are not growing from the minimal media. I need to supply either ornithine or citrulline or arginine to grow. Why do I need to give ornithine to them to grow? If all the enzymes are working, that is E2 and E3 is working.
So they can go through the whole process.
That means they cannot convert ornithine from minimal media. That's why I need to supply ornithine from outside. They cannot convert ornithine from minimal media. So that is another step. Another step is blocked. That enzyme is blocked. That's why I need to supply ornithine from outside. Okay? When I need to supply ornithine from outside, that means they are mutant. They cannot synthesize ornithine from minimal media. Now from this chart, we can determine what are the steps from here. Can we? If I need to give ornithine to them, they can grow. So if I need to put ornithine here, they can grow. That means these two processes are okay. If I need to give citrulline from here, they can grow. So these processes are okay. Now I can check in this way. For this group, I need to put ornithine, I need to put arginine only, so they can grow. They cannot grow in citrulline or ornithine. So that means arginine is the last one for their growth. Citrulline and ornithine is in between. Okay. From here, we can say that if I put citrulline, they can grow. They need arginine to grow. So citrulline to arginine is the one-step process. So citrulline is in the middle of this process, but they cannot grow in the ornithine. If ornithine is here in the middle, if I put citrulline here, suppose here is citrulline, here is ornithine. Okay? So then the process would be ornithine to arginine. So if I put ornithine here, they will grow, definitely, because enzyme 3 is working in such case. But here is the question is that when we are putting citrulline here, they can grow. That means citrulline is in the middle. Clear? Just think the logic. Why? Why not? Why? Why not? Okay? So in such case, we know that, so we have created the mutants here. And those mutants, we know that we irradiated with X-ray, so we created mutants and we created 3 gene mutations here. Okay? And this process requires 3 enzymes to synthesize arginine for their growth. And from this chart, we know that the defect is here in the enzymes. Okay? Now we created 3 mutations and 3 enzymes are defective. So can we see that that gene is creating that enzyme? Our main basic thing is that we did the mutation. Mutation is in the gene. And after that mutation, when we tested this one, we thought we saw these things. These are the substrates or the products, whatever it is. We can give it from outside, but they cannot synthesize that one. That means that enzyme is defective. We have created the gene defective and we are observing that enzyme is defective. Can we say that one gene is equal to one enzyme or one gene is producing one enzyme? Yes or no? Again, think logical. These are the products. I can supply it from outside, so there is no problem. Why they cannot grow? Because their enzymes are defective. Enzyme 1, enzyme 2, enzyme 3 for 3 different groups, not only in single group. 3 different groups, they have 3 kind of defective enzymes. I didn't defect all those enzymes. I didn't do that. What I did the genes. I disrupted the genes there. I mutated the genes there. I didn't do anything with the enzymes. But as a result, I have found that the enzymes are defective. Can I say that that gene, what I mutated, that is creating one of these enzymes? Yes or no? Logically. Think it clearly. Yes. Think it clearly. I mutated the genes. I didn't do anything with enzymes, but as a result, I have found that enzymes are defective. So can I say that one gene, that what I mutated, that was creating that enzymes, but as I mutated the gene, that cannot create that active enzyme? Yes? So that means now I can say that one gene can produce one enzyme. One gene can synthesize one enzyme. Clear? Still, if you have any questions, just ask me. But make it clear. Totally logically. Logical explanation is very, very important here. Otherwise, it is very difficult to understand. Okay? Now go to another part. This problem, I put it here. I don't want to discuss because we don't have too much time. Do it without seeing the answer. Okay? Do it by yourself. Practice it by yourself. Then match with the answer. Okay? Think by yourself. Do it logically. Just draw this diagram and try to find out what gene, what is mutating, what enzyme is mutating. Then check the answer. Okay? Now, we know that. So, from here we know that one gene is disrupting one enzyme. That's okay, that's fine. Now, what is enzyme? What is the chemical property of enzyme? Carbohydrate, protein, lipid, or nucleic acid. There are 4 things. Carbohydrate, protein, lipid, nucleic acid. The macromolecules. What is enzyme? Protein or carbohydrate? What do you think? What do you think? Didn't you do the protein structure, right? Before? I think so. Yeah. So enzymes are proteins. All the enzymes are proteins, and enzymes are very, very specific. For each and every step of biochemical process, one specific enzyme is required. Okay, and their chemical properties, they are proteins. Okay, so from our previous experiment, Neurospora experiment, we have found that one gene is synthesizing one enzyme. One gene is synthesizing, so one gene is equal to one enzyme. Now I am changing this part because enzymes are proteins. So logically, one gene is equal to one protein, right? Because enzymes are proteins, so one gene is equal to one protein. Now the question is that protein has four kind of structures: either primary structure, secondary structure, tertiary structure. This is one polypeptide chain of different shapes. It is linear. This is sheet-like structure having hydrogen bonds there between them. And this is three-dimensional structure with the sulfide bonds. So hydrogen bonds are there as well as the sulfide bonds. So one polypeptide chain can be of linear, two-dimensional or three-dimensional structure. When we are going to quaternary structure of the protein, we know that not only a single polypeptide chain, couple of polypeptide chains are together. Okay, this is making the quaternary structure, different subunits. Now the question is that one gene is equal to one protein. One protein can be made up of couple of polypeptides according to this quaternary structure. This protein can be made up of couple of polypeptides. So what does it mean? One gene is equal to one protein as a whole. That means one gene is creating all those polypeptides together, or just a single polypeptide? Now go to deeper, little bit. One gene, one enzyme, that was fine. But I now know that one gene enzyme is equal to protein. So one gene is equal to one protein, logically. Okay. Now I know that protein is made up of a couple of polypeptides, not a single polypeptide. Now the question is there, is that gene is creating all the polypeptides or not? So that was the question. Okay. Now we need to solve this question, and actually one scientist, a group of scientists, they did one experiment, this one. So for this one, we know one disease is called sickle cell anemia. Do you know that? Sickle cell anemia? What is that? Sickle cell anemia? The blood cells are sickle set like this. What is the result for this one? Result means that if I have this kind of blood cells in my body, what will happen to me? What is that? What is the function of blood cell? RBC. This is RBC, red blood cells, not the white blood cells. Oxygen deficiency. Main thing is that oxygen deficiency, because the surface is less. For the less surface, they can have oxygen deficiency. Generally, those people who have sickle cell anemia, that's why it is called anemic. That means having oxygen deficiency. They have some kind of respiratory problems. Okay, and that is the result of sickle cell anemia. And this sickle cell, actually the hemoglobin is controlled by a kind of gene, and that is called HB or hemoglobin gene. So HB plus, this is the wild type. Okay. So in one person we can have HB plus, HB plus. Why do I need to write HB plus, HB plus? No, these are dominant. But why do I need to write twice? I asked this question in my other biology class also. Two genes, why?
One from the mother, one from the father.
That means what? What is the term here? Just one word. One word. What is the gene? What is that things? At first, we are diploid organism. We are a diploid organism. That means having one set of chromosome from our father, one set of chromosome from our mother. But what are those chromosomes? What is the word? Autosome means the somatic chromosome, and sex chromosome means the chromosomes determining sex or gender. What are those chromosomes? Again, the meiosis. Again, the meiosis. What are those chromosomes called? Homologous chromosomes. The simple term is homologous chromosomes. Okay. So we are diploid organism. In homologous chromosomes, these are two alleles. Two alleles. If it is this one, then this person is normal. Okay. If this person have this kind of gene, HBS and HBS, HBS for sickle cell, so this person is sickle cell anemia. Okay, this is totally anemic, sickle cell anemia. But this is recessive. This one is recessive, this one is dominant type. So we can have one heterozygous condition, right? The heterozygous condition is HB plus and HBS. This is called sickle cell trait. Sickle cell trait. Normal, sickle cell trait, and sickle cell anemia. These are three groups. Okay. Now the thing is that this sickle cell and those kind of these kind of persons, they can have a kind of normal cells as well as sickle cells. Kind of 50-50 population, something like that. Both the normal and sickle cell, and they have little bit respect. This hemoglobin consists of lot of mainly two polypeptides. Okay. And this difference between this HBS gene and HBS protein, HBS polypeptide or HB plus polypeptide, they Ingram actually did this experiment. This is called the chromatography. So they took the blood of or isolated the protein, HBS protein and HB plus protein, and they did the chromatography. After chromatography, they have found that only these polypeptides is different in both groups. Only this polypeptide is different. These are polypeptides. All the polypeptides are there. Only this polypeptide is different. Okay. So what is there in this polypeptide? This is the normal polypeptide for the hemoglobin chain, and this is a sickle cell polypeptide. The sixth position here is valine, and in the sixth position there is glutamine. Only one amino acid change. That one change in amino acid can change the whole polypeptide, the property of whole polypeptide. And this is actually the sickle cell structure, original structure, original RBC of sickle cell. And this is normal RBC. So only this change, this whole polypeptide change can create this kind of disruption. Okay? And this is mutated. This is the mutated one. So that mutation caused the change in the polypeptide chain. Okay? Now can I say that one gene is equal to not one protein, but one gene is equal to one polypeptide after this experiment? Because the change in only one polypeptide, not the whole protein. Only one polypeptide chain is different, and that is created by one gene mutation, that is the hemoglobin S gene mutation. So now can I say that one gene is equal to one polypeptide? Okay, so these are two, that is most popular experiments from where we can decide that one gene is equal to one enzyme. That is the first thing. Now the whole thing, one gene, one enzyme hypothesis is now one gene, one polypeptide. So one gene, the function of one gene is to create one polypeptide chain. Okay, That is called the expression of gene. The gene is expressed means what? The gene can create one polypeptide chain. That's why it is expressed. Okay? Now, this expression expression of genes means, so now I can postulate that central dogma hypothesis again. What is central dogma? Central dogma. What is that? Who proposed that? Central dogma. This is the very common things. You need to remember all the names. What is central dogma? Central dogma hypothesis. Who created that? Who postulated that hypothesis? So DNA can be replicated. From DNA, mRNA is produced. From mRNA, the protein is produced or synthesized. This is the central dogma hypothesis proposed by.
So for the midterm, you know the name of who created what?
A little bit.
Okay.
These are very common things. You have to remember, because if you cannot say who proposed the central dogma hypothesis, it is really very bad. I can say that. If you don't know who actually demonstrated the structure of DNA and you are a genetics specialist, then it is really very bad. It is really very bad. And those two persons are very, very popular in genetics. You need to know two names at least in genetics. One is Gregor Johann Mendel, and another is Watson and Crick. These two persons. So Watson and Crick actually they demonstrated the DNA structure, and based on that structure, what the whole molecular biology, whole biotechnology is based on that one, and the whole genetics is based on Mendel. That factor you need to know this. Anyway. So this is proposed by Watson and Crick. So the first thing is that DNA to DNA is called DNA replication. DNA to mRNA, that is called transcription. And RNA to protein, that is called translation. Okay? This is our whole thing. Transcription and translation. Here between this one, this full translation and mature mRNA, that is called the RNA splicing. And here also there is another short part that is called post-translational modification. With the mature protein. Okay. In biotechnology, we have some kind of reverse process. That reverse process is from here to here, that is mRNA to DNA, that is called reverse transcription. These are the modification. Reverse transcription. Okay. These are the modification of original central dogma hypothesis. Now we know these things because some bacteria, they can create DNA from their RNA. We know that is called reverse transcription. Using that reverse transcription, we can create cDNA library from mRNA. Okay, using the enzyme reverse transcriptase. So right now we need to talk about this part, the translation. Translation means translated into different language. Up to this, the language was same, only the nucleotide language. Right now, the language will be changed from RNA to protein. That means language from nucleotide to amino acid. Different language. English to Greek, different language. It is like that. Okay? That's why it is called translation. Okay. Now we know the structure of mRNA. What is mRNA? mRNA is nothing but a nucleotide structure made up of chain of different nucleotides. And here, other than T, we have the U. Suppose like G, C, C, A, U, like that. Okay. Other than thymine, we have uracil, and that is the nucleotide structure. All the nucleotides are there. Okay, now the question is that how the amino acids are produced. Okay, they can be coded. They can be coded from here. Now, what are the codes? That is called the genetic code. Okay, so we need to think about this code and the codon structure and different types of codons here. What are codes? What are the codons and different types of codons? What is the start codon? What is the termination codon and those kind of things? And whether this is universal or not. Whether in bacteria there is same code, whether in eukaryote there is the same code or not. Okay. Now the problem is that we have only 4 things, 4 nucleotides, A, T, C, G. Those are 4 nucleotides. And we have 20 amino acids. So 4 nucleotides and 20 amino acids. So how can 4 nucleotides code for 20 amino acids? If suppose adenine code for 1 amino acid, then adenine need to code for so many amino acids there. And that is a disruption. The total, if I do it mathematically, so there are 4 nucleotides, they can code for 16 amino acids as a whole total together. If I think if it is 1, they can code for only 4 amino acids. If I think that 2 nucleotides together can code for 1 amino acid, that is this one. So we can have only 16 amino acids. 20 amino acids are not covered here. If 3 codes, 3 nucleotides together can code for 1 amino acid, we can have 64 codons. So 1 codon can code for different amino acids or same amino acid. That can be possible, but we can cover 20 amino acids anyway. But from there we cannot cover the 20 amino acids, only the 16. So this was first thing that probably the 3 nucleotides code for 1 amino acid. This was hypothetical first. But finally, Francis Crick, actually in 1961, he actually decoded this thing that really 3 amino acids, 3 nucleotides code for 1 amino acid. Okay, that is true. Now, in these 64 codons, there are a couple of things. One is called stop codon. 3 are stop codons and 61 are sense codons. Sense codons means they can sense for amino acids. Okay, so this is the hypothetical structure of codons. That means one codon is made up of 3 nucleotides, can code for 1 amino acid. So what is a codon? Codon means made up of 3 nucleotides, can code for 1 amino acid. It can call for 1 amino acid. That is called a codon. Okay. Now, so the thing is that we have 64 amino acids, 64 codons and 20 amino acids. That means 1 codon can call for different, or so many codons can call for 1 amino acid. 64 codons call for 20 amino acids. So a lot of codons can call for 1 amino acid. That can be possible. Okay, that is the first thing. That is the degeneracy of code. Now, some codons, that is called the synonymous codons, that specify the same amino acid. That is, suppose 6 codons together can call for one amino acid. So they are these 6 codons, they are called the synonymous. And there are 3 most important things. Always the initiation codon or the start codon is AUG. This is the start codon, and you need to remember this code. Okay, what is the start codon? AUG. That is the only thing in the whole biosphere. In any organism, the start codon is AUG. Main start codon. Right now we know that couple of other start codons are there, but those are not really very important. Main is AUG. And there are 3 termination codons or stop codons, that is UAA, UAG, and UGA. These 3 are called stop codons. Okay. So this is our whole genetic code. So how can we determine the genetic code? At first, UCAG, I put all those things there. For each row, we put UCAG again. For each row, we put UCAG again. So these are the 3 systems. So that is U, this is G. So that is U and G here. And here is another U. Okay. So UUG or GUU is that way. If I start from this way, so that is G, U, and U. G, U, and here is C. So GUC. Again, G, U, here is A, so GUA. This way we can actually demonstrate the whole codons, 64 codons, and these are all amino acids here. So all these 4 codons, they can call for valine. Okay, all these 4 codons, they can call for alanine. These 2 call for aspartic acid, these 2 call for glutamine. Okay, only this one called for methionine. Only one thing, that is the start codon AUG, it calls for methionine. And that is the only codon calls for only one amino acid. Another is there, that is called the lysine, I think so. Okay, tryptophan. UGG. That is only one codon call for only one amino acid. Other than that, a lot of codons, mostly 2, 3, or 4 codons, even the 6 codons, they can call for one amino acid. Okay. Now the question is that if these all 4 codons call for one amino acid, then the condition is that, okay, at first we need to know how they call for the amino acid. One RNA is required, that is called the tRNA. We already went to the structure of tRNA. The tRNA has anticodon part. This is a normal structure of tRNA, suppose like that. It is anticodon means here is U, here is A, and here is C. Anticodon means just opposite to that codon. If they have this anticodon structure, they can bind with the codons. Okay. Now for tRNA, so if these are the codons, tRNAs can have these anticodons as well. All these 64 anticodons. Think about that again logically. tRNA is responsible for bringing the amino acid here. This is the carrier, or the bus, or the public transport. That is the public transport, and that public transport can go to the particular bay. I can stop here. I can park in this bay only, not others. Okay, this is the codon anticodon structure. So I need to determine that in which parking lot I need to go. Okay, if I am the driver of this carrier, I need to figure out that which parking lot I need to go. Okay, this is my signal, so I will go to this one. Another one is seeing that, okay, that is my signal, I will go to that one. Okay, I am bringing the amino acids here. So that code is calling, okay, this is your signal, this anticodon can come to this code only. Okay, so in such case, for all these codons, we can have 64 anticodons. Otherwise, they cannot bind with that one. We can have 64 anticodons. Am I right or wrong? Think logically. We have 64 codons. Okay, tell that stop codon. For stop codon, there is no tRNA. So 64 minus 3, that is 61. We must have 61 tRNA with the different anticodons so they can bind with that one, right? Am I right or wrong? Otherwise, they cannot bind in this way, right? But we have only 51 tRNA. We have only 51 tRNA. And we have 61 codons. So how can it be possible? That binding, that kind of binding. If it is so restricted here that only codon and anticodons can bind, then how can it be possible? Is it possible or not?
Multiple codons call for.
Yeah,
some
multiple
amino
codons can call for one amino acid. Suppose this one and this one is anticodon is G, U, A. This is the anticodon. It will call for another amino acid here, amino acid 2. Or it can call for the same amino acid again. So like C, G, G, it can call for same amino acid again, like A1, something like that. Just think like that. So amino acid and tRNA, that is not a problem. Problem is that we have 51 tRNAs and we have 61 codons. So particularly for some codons, there is no tRNA at all. And we know only there are 3 stop codons. For those 3 stop codons, there is no tRNA available. We know that. So how is it possible? How this pairing can be possible? That is called Ubel hypothesis. That is the main thing called Ubel hypothesis. And what is Ubel hypothesis or Ubel pairing is that main 2 is responsible. These 2 are responsible for binding. For each codon, the first 2 pairing is most important. Third one can be of different. G can pair with U. That is a very less binding there. That's why it is called Ubel hypothesis. Okay? The main thing is that only these 2 is very, very important. The last one is not so important. In such case, they can pair with this codon and they can bring the amino acid here. This is called Ubel hypothesis. So 51 amino acid can bind with all 61 codons in such way, if that Ubel pairing is there. Clear? And this one also proposed by Francis Crick. He is the pioneer of almost all this part. And Watson was his professor actually. So if you are interested later, I will suggest you Francis Crick's, the book is called Molecular Biology of the Gene. And you can just, but the size is like this. This is the size. But if you can go through that one, another very interesting book is Benjamin Levin's Genes. Actually, I have the genes 5. Right now we have gene 12 or something, but gene 12 is thinner. Gene 5 is like this size. And their DNA replication is almost like 175 pages. Okay? If you are interested, you can go through that one time. In your whole life, you will not, that is, forget about this. And in biotechnology, actually in modern biology, all these things are very, very important. Next class or next couple of classes, we will do this one. Let's tell me this one. Through Ubel, a single dash can pair with more than one dash. What do you think? question is that not tRNA or amino acid. tRNA is not binding with the amino acid here in this way, codon-anticodon pairing. That is not true. Group of 3 nucleotides in DNA, codon in mRNA. Codon is not in mRNA. In mRNA, or not nucleotides with codon in mRNA. That is not true because it is the pairing with tRNA and mRNA. So now the question is that codon anticodon or anticodon codon? So you say that true, well, a single codon can pair with more than one anticodon? Or a single anticodon can pair with more than one codon?
The anticodones will that carry the trans.
Yeah. What do you think? A or B? Anticodon can pair with different codons. Anticodons can pair with different codons. Okay, now there are certain things. Now, what is the main thing is that, okay, we know that 3 nucleotides is one codon. Okay, so these 3 is one codon. What about why not these 3 another codon? Could be possible. Why not? 3 nucleotide is one codon. So the first 3 AUG is one codon. The next one is UGC. The next one is GCA. That can be possible. Or suppose like it is disruptive, like AUG is one codon, then GCA is one codon, then this EU is not counting, then GCC is another. But truly, they are following a very strict rule. All 3 codons are like that. They are not overlapping, and there is none is skipped there. Okay, that is called the reading frame. So reading frame is always 3 codons. They are non-overlapping, and they are generally universal. Universal means in prokaryotes and in eukaryotes, these codons are same, almost same. In case of eukaryotes, we have some kind of different codons for the start codon, that is CUG or something like that. Sometimes they can start the synthesis, but still the AUG is the main start codon. Okay? They are nearly universal. So these are some exceptions of universal codes. You can go through that one. Okay, now let's go to the translation process. So what is translation? Translation is that we must have one mRNA here, and these tRNA, they can bind with the amino acid, they can bring the amino acid here with the proper codon, and these amino acids should be bound with each other to make the polypeptide chain. In a simple way, we can think like this. Okay? So the first step is that they need to start that one. The first thing is that the binding of tRNA and amino acid. This is the first part. Binding of tRNA and amino acid. That is the first part. And that is happening in the cytoplasm. No mRNA is required. There. Okay. The second thing is that binding of this whole thing. That is the starting on initiation. Initiation requires one thing, one main component, that is called a ribosome. Okay? And the whole protein synthesis is inside ribosome. And it is either in endoplasmic reticulum or in cytoplasm, free ribosome or the attached ribosome, but the protein synthesis factory is within the ribosome. So for initiation, the one thing is that we need to think about that for initiation, something is required to put the ribosome or put the mRNA into particular ribosome, fit that codon into particular space, and call for the tRNAs there to attach the amino acid. Okay. Then the elongation of the chain, so they need to read that ribosome need to read all the codes here until the termination codon and need to attach all the mRNAs to that, all the amino acids onto that. And finally the termination with the stop codon. So this is the whole process here. So starting from the AUG, ribosome is attached there, then they can go through this mRNA chain from 5' to 3' position, and as a result, they can synthesize the whole polypeptide chain. This is in a nutshell, the whole thing. Okay. So the first part is that charging the tRNA. What is the charging of tRNA? So tRNAs are there in the outside in such way, like that. And amino acids are there. They have the definite anticodons, and that tRNA is specific for particular amino acid. Okay. Each and every amino acid is specific for a particular tRNA. If one amino acid is called by 6 tRNAs, so those amino acids are very specific for all those 6 tRNAs. Okay. This binding of tRNA with amino acid, this binding needs a particular enzyme. There. This is called amino acyl transferase, that enzyme. And this thing, that is the point, after binding with tRNA and amino acid, this is called amino acyl tRNA. Okay, so at first tRNA was there, amino acid was there, tRNA binding with amino acid by the enzyme amino acyl transferase, and as a result, a compound is formed with tRNA and amino acid, that is called amino acyl tRNA. Clear, all those terms? As there are 20 different amino acids, there should be 20 different amino acyl synthetases. Okay? For each amino acid, there is one specific amino acyl synthetase, or it is called transferase also. So this is the way they can bind with that. tRNA has this kind of clover-leaf structure. This is the anticodon site, and that is the amino acid binding site at the 3' region. That is a slight overhang there, and there actually the amino acid is binding. Generally, one A is there, one adenine is there, and it is binding with that part. Okay, this is not codon anticodon sequence. This is the binding with amino acid and tRNA. And these are the steps here. The binding with amino acid and tRNA, one molecule of ATP is required, and this energy is required to bind this one and making the amino acid tRNA. This is, I just put the detailed step here, but that is not really very important for you. So you can just know that amino acid is binding with tRNA by amino acyl synthetase, and one molecule of ATP is required for that one. That is the binding. That is the first part. Now, amino acids bind to which part of RNA? Where is tRNA? Anticodon site or not? Try to figure out that picture. That is tRNA and amino acid. Not tRNA, mRNA, not codon anticodon sequence. tRNA and amino acid. That is the end tail there, and that is the 3' tail. That 3' tail, the amino acid is attached. Okay? So it should be 3' end. Okay, now that is one part is gone. Now we need to think about the ribosome. Ribosome is very, very important. Okay. Ribosome has two units. One is called the small subunit, another is called the large subunit. Okay? These two subunits are separated at the beginning. During protein synthesis, these two subunits are together. In case of bacteria, this whole thing is 70S, this one is 30S, and this one is 50S. In case of eukaryotes, this whole thing is 80S, this one is 40S, and this one is 60S. Okay? There are two subunits. In these subunits, there are certain chambers. One is called A site, P site, and E site. A site is called the amino acyl binding site. So new tRNA with the amino acid can bind with this part. This is the entry part. Okay, there is certain chamber here, and the tRNA with amino acid can bind to this part. P site is called peptidyl transfer site or peptidyl site. What is peptidyl site? When the mRNA is already formed here. So what will happen? In the A site, first tRNA will come, they can bind with this one, then this whole chain will transfer to this one. Okay? Then this whole ribosome will move to another codon. So this A part will come to the P part, and this part will go to the E part. Okay? If I move just one codon, this one, just transfer one codon. So this thing will come to here, this thing will go to here. This is called the exit part. From here, this tRNA will be released, and the chain is already transferred to here. And this one will go to P part again. So another one will come here. This chain will move to this one. This ribosome will move to another codon. This one will go to the exit site. There is no chain there. It will be released, and this one will be here. This is the way the whole procedure is going on. There are three sites. The first is called amino acyl site. P site is a peptidyl site, and E is a exit site. Attachment of new tRNA with amino acid, transfer of amino acid into that one, the existing chain, and this is the exit side of tRNA. Other than that, there are two main areas also. These are three main parts of the ribosome. There are two main areas. One is called the decoding center. This one is called the decoding center, and that one is called peptidyl transferase center. Okay? Decoding center means this here, it will decide whether that tRNA will sit here or not. It is the perfect tRNA or not. Who will call that? Every tRNA is coming there and trying to bind with that one. So this part, actually this ribosome is made up of rRNA. So that rRNA will decide whether this tRNA is matching with this codon or not. And if it is matching with this codon and decodon is matching, then it can sit here, otherwise it will not sit. That is called the decoding center. It will decide whether that tRNA will sit here or not. Okay? And peptidyl transferase center, the function is that need to move the whole chain, the pre-existing chain with the new amino acid. That is the forming the peptide bond there. Forming a new bond, new peptide bond, because both amino acids are joined together by a bond called peptide bond. So it is forming the new bond there and transferring the whole chain into that. That is called the peptidyl transferase center. So mainly there are three chambers. One is A site, amino acyl site, peptidyl site, and exit site. And there are two other short areas, which is called the decoding center and peptidyl transferase center. And these are all inside the ribosome. Clear? Now, what is initiation? Initiation needs one type of codon, that is the AUG. That is the main codon, that is called the initiation codon. Okay? During initiation, And particularly, there are two sequences we knew that in transcription. One is called the signed Dalgarno sequence in prokaryote. The same type of sequence in eukaryote is called the Kozak sequence. It is in prokaryote, that is signed Dalgarno sequence, and in eukaryote it is Kozak sequence. The sequence is different. Signed Dalgarno is not present in eukaryotes. Okay. What is the function of that one? Ribosome is made up of rRNA. This is a part of rRNA. They can just adjust that mRNA to fit this AUG codon in the first P site. This is the function of signed Dalgarno or Kozak sequence. They can just adjust the first start codon into the first P site. In short, this is the function of this one. Okay. Now, for this initiation factor, we need couple of things first. One is the mRNA is required definitely. Without mRNA, there will be no decoding part. Small and large subunits of ribosome. It is required, but the ribosome is the main factory there where there will be all the transfers and other things. A set of 3 proteins called the initiation factors. In case of prokaryote, there are sets of 3 proteins or 3 initiation factors. In case of eukaryote, there are 12 initiation factors. Okay. Initiated tRNA with N-formyl methionine attached to it. In case of bacteria, the first codon, first codon is AUG, and that methionine is formilated. A cap-like structure. It is a little bit different methionine with the formilated methionine that is called F-met in case of prokaryote. In case of eukaryote, it is methionine, simply methionine. Okay. And guanosine triphosphate for the energy. For all this transfer, they require energy, and then 1 molecule of GTP is required there. So these are the main things required for initiation. Let's go to the next one. So what is happening here? We have 2 units of ribosomes there, that's fine. We have the mRNA here. mRNA signed Dalgarno sequence, it will fix that AUG portion in the P site of the small unit of ribosome. First, the small unit of ribosome is there. And initiation factor, it is helping that ribosome to fit into that site. Okay. So that AUG codon is fit into the P site here. And signed Dalgarno sequence or Kozak sequence, they are helping them to fit in the proper position. Okay. Then the first thing is that the F-met methionine, that is the anticodon is UAC. If it is AUG, the anticodon is UAC. That tRNA is with this F-met methionine. They can come together and they can bind it with the P site. Okay. Can come here and then bind with this codon, you can attach here. And for this part of the IF2 and GTP, they are required. They are helping that tRNA to come here and attach with the mRNA. First starting mRNA, start code. Okay. So the first thing is that small unit of ribosome there. Start codon AUG is fit into the P position or P chamber of this small unit of ribosome. F-met methionine is coming here, that tRNA, and binding with this AUG codon. Still that subunit, small subunit is here, not the large subunit yet. So this is the second step. Third step is that now we need the large subunit. Now the large subunit is coming here and joining to that one, so the whole ribosome is formed. So after this initiation, the whole part, what we can see that AUG codon in the P site of the whole ribosome. Whole ribosome is formed with the large subunit and small subunit. At the P site, the AUG codon is there, and F-met methionine with the tRNA, it is attached with that one. Now the initiation is complete. We don't need initiation factors anymore. Initiation factors, they are helping to set these things, and the signed Dalgarno sequence or Kozak sequence, they are helping to fit these things in the proper place. Now their function is done. We don't need that. Initiation is done. Okay. Now what will happen? In case of eukaryote, it is a little bit difficult because eukaryotes have one cap here. So there are a lot of gap binding proteins are required. In case of Kozak sequence, initiation factors, and they need this cap binding protein to fit the ribosome into the proper place. Okay. All of these things. And the poly-A tail is also helping with this one. Okay, so this is our initiation. After initiation, the A chamber is open here, and we know that in the A chamber, new tRNA will come and attach to the A chamber. So new tRNA here is CCC. So GGG is taking the proalanine amino acid. It is bringing there, attaching with this one sequence. Okay. Now, this F-mate amino acid will bind to the pro-amino acid. Right now, it will be transferred. That is the peptidyl transfer. Both the tRNAs are here. Now the amino acid will be transferred to that one. Okay, forming the bond. So they are forming the bond, and the amino acid is transferred to the new tRNA with amino acid. Ribosome moved one step. So that one was in A site. Right now it came to the P site. That previous tRNA was in P site, now went to E site, and exits. Clear? This part? And this way, the elongation is more or less simple, just like new amino acid on the A site. Here is the P site, the previous amino acid were there. This amino acid will transfer to this one. Ribosome will move to one set, and then this tRNA will go to exit site. It will release, and another new one will come here. Just like this way, the elongation is going on. Now, what will be the termination? The stop codon, there is no tRNA for the stop codon. But that stop codon is recognized by one factor that is called the release factor. Release factor can recognize the stop codon. When the A site of the ribosome is in the stop codon, then there will be no amino acid. The decoding center, they will call for the amino acid, but there is no amino acid. Then the release factor can come and attach to that part with the stop codon. Function of the release factor is that releasing everything, the whole unit, the large and small subunit, the mRNA and the polypeptide chain and the tRNA, everything they will release. This is a simple termination. Only the release factor is required for that. Now,
in translation, amino acid are assembled into proteins. In translation, amino acids are assembled into proteins. Bacterial translation is initiated in 3 steps. First, the small subunit of the ribosome binds to the mRNA so that the P site is positioned over the start codon. Second, the initiator tRNA with N-formylmethionine attached binds to the mRNA through base pairing between the codon and the anticodon. Third, the large subunit of the ribosome joins the complex to complete initiation. During elongation, amino acids are joined to a growing polypeptide chain. The first step of elongation is the delivery of tRNA and its amino acid to the A site. In the second step of elongation, a peptide bond forms between the amino acids. The third step of elongation is translocation, the movement of the ribosome down the mRNA. After translocation, the tRNA formerly in the P site is now in the E site, from which it moves to the cytoplasm. Now the elongation cycle repeats itself. A tRNA and its amino acid occupy the A site. A peptide bond forms, and the ribosome translocates to the next codon. As the cycles of elongation continue, the polypeptide chain grows by the addition of amino acids to its carboxyl end. The last stage of translation, called termination, takes place when the ribosome reaches a termination codon. Release factors bind to the termination codon, causing the release of the polypeptide chain. The ribosome then translocates one last time. The tRNA and release factors exit the ribosome, and then the ribosome dissociates. Remember that several accessory proteins not shown here also participate in translation.
Clear? I think this is more or less simple. Okay. Okay, so we have the summary part here. That is the same thing in the video. And just a couple of things here, that one ribosome is not always working on the same mRNA. If it requires more protein on the mRNA, lot of ribosomes are acting on that. And that structure is called a polyribosome. Okay? From one mRNA, lot of proteins are formed. Same protein are formed by different times, but at the same time, almost like similar. This is called the polyribosome structure. One mRNA with several ribosomes. This is one thing. And what will happen after that? Protein is formed, and that is the first polypeptide chain. Okay, so each protein, there should be the first amino acid should be the methane. Am I right? As AUG is the start codon, the first amino acid of each and every protein will be methane. Am I right? But can we see that? No. That is the thing, is that there are some other post-translational modifications are there. And not only that, proteins are bound with, suppose hemoglobin. One heme is there, that is iron compound. In some protein, there are some carbohydrates are there, some kind of lipids are there. All these modifications are needed after the formation of this polypeptide chain. The first couple of amino acids are called the signal peptides, signal peptide. And it is recognized by signal peptidase enzyme. That signal peptidase enzyme can bring that polypeptide chain into endoplasmic reticulum. There, the signal peptide part is cut out, and a little bit modification is there. The first primary structure to secondary structure, secondary to tertiary, tertiary to quaternary. Then it is kept in the endoplasmic reticulum. That is called the post-translational modification. And not only that, in the Golgi bodies, when it is transferred from endoplasmic reticulum to Golgi bodies, in the Golgi bodies, there are some other modifications also. The addition of other protein with that, addition of carbohydrate with that protein, and then it is fully packaged protein right now. From Golgi bodies, it is transferred to other areas. That is the USPS system. Packaged protein is there, everything is required, all the manual and everything, the whole structure is there. Now it is ready to go outside the cell to function there. Suppose like antibodies. Antibodies are producing inside the cell, but it is going to work in some other part. Hormones, it are producing inside the pituitary gland, but it needs to work in kidney. So the pituitary gland cells, they are packaging this kind of proteins, they are creating these proteins, they are packaging these proteins, and they are transferring these to other part of the cell. And not only that, another thing is that if the protein is not required, then this ubiquitination is occurred there. that in this part, lysosome, ubiquitin is added to that one in the protein. So lysosome can find out that this protein needs to be degraded. Okay, this kind of protein modifications is there in a very simple step. That is after production of protein, the first couple of amino acids, they are called a signal peptide. That is characterized by signal peptidase. Then it goes to endoplasmic reticulum for further modification, secondary structure, tertiary structure, quaternary structure. Then it packaged to Golgi bodies, and in the Golgi bodies, the addition of carbohydrate and proteins or lipids, it is happening there because in case of cell membrane protein, phospholipid structure, and some integral proteins are there. So this kind of modification is there, and then the protein is fully packed. Okay. This is more or less all about translation. Clear? That one? Next day, we'll go for a little bit, these two chapters. We had two chapters, the prokaryotic and eukaryotic modifications, that is the gene regulations there. But I will combine these two chapters in a very short form in the model because there are a lot of things and you really don't need all this. Okay. I will just go for the Lac-Operon model, Lac and TRP model, and one eukaryotic model. That is. Okay, and go for your test. Really, there is no such on transcription, but study the replication part and all other things from beginning. But you have 3 chances. It's not a problem. I hope you will score.
3 chances are better.
All of you. Okay.